Let me introduce Dr. Emmanuelle Maguin, who's a senior researcher at INRAE, Paris-Saclay University, working on diet, microbiota, and host interaction. Please, Emmanuelle.
Thank you, Juan and Moez, for this nice introduction. I'm very pleased to be here. I came to ONIRIS years ago when I was head of a research division, coming to visit some laboratories which were associated to INRAE. And I'm very pleased that we could, in the team, develop this collaboration with ONIRIS and the team of Juan Hernandez. I think it's very fruitful and very nice to be here. So I'm going to tell you a little bit about nutrition and human microbiota. And I don't know why you're doing that. Is it okay? Yeah. And I will start with just to remind you what happens with our agri-food system in the last 70 years or so. It changed a lot. There were lots of transition. And basically, we used more and more chemical inputs. There were a large program on genetic selection, globalization of the whole system, and industrialization. And all of that lead to agri-food systems which are right now contributing to climate change. It's estimated to be about 30% of climate change modification. So those modifications also change the food offer, meaning that the food safety increase, which is very, very good, food availability too, and globally the yields of primary production increased. But on the other hand, we decrease the diversity of the food that we are eating. There were development of lots of new products and new processes, and including ultra-processed food, and we know now that it's not so good for our health. And the exposome, meaning the chemicals to which we are exposed through food system and through our nutrition, increase really dramatically over the last century. And in addition, the cost of food is not so, I mean, it depends, but of the country, of course, of the world, but the share of the budget, of household budget, which is spending on food, increased over years. Of course, those modifications also impact the food demand and the consumer behavior. And both kind of, there is this kind of interplay. So now, in our countries, consumers change their diet over the last 60 years. And we switch from, 2 million of human diet based on very fiber-rich diet to low-fiber diet. And today in France or in Europe, most of the people, on the average, we are consuming 12 grams, 20 grams at most of fibers in our diet, which is far below what was happening before. So the fiber intake decreased by at least 8-fold. We increased a lot the consumption of simple sugar, digestible sugar, animal proteins, lipids, salt, and we also decreased our consumption of fermented food. And it's a very, 50 years in terms of genetics and adaptation of our body is very short. It's a very limited time. So we had no possibility to adjust ourselves, our body, our microbiota, our physiology to those kind of transformation. The consequence is that we have this uncontrolled rise in incidence of chronic non-communicable diseases, which is the main cause of death worldwide. And it's not this year. It was already the case in 2019, and it has a tremendous burden on our health system. And I just put this figure here because you see, for example, malnutrition. So there is 3 aspects in malnutrition: undernutrition, overnutrition, and micronutrient deficiency. And it changed considerably over the last 20 years. And now, as you can see in this map here, obesity and nutrition or malnutrition problem concern all countries worldwide. It's not, it's not overnutrition. Obesity is not specific of rich country. It's everywhere. And malnutrition is everywhere also. Okay, so we are really in a situation where those non-communicable diseases are very important to consider and that we have to develop solutions. So, what is the link with microbiota? So first, I just want to remind you that a number of studies, and at that time it was really a large study, those thousand or few thousand of people studied for their microbiota, their lifestyle, their nutrition. So there were those 3 studies which were kind of pioneer at this time, who tried to identify the factors that are shaping our microbiota. And here are what they found, and we know that there is other factors which are unidentified yet, and which are probably important. And it explains probably because why we have some difficulties in the field. So of course we have birth condition and diet, from birth to the end, I would say. Medical treatment, it's clear that antibiotics are targeting bacteria. So when we have antibiotic treatment, we are targeting all the bacteria which are living with us. Physical activity, the environment, whether you are living in urban or rural environment, whether you have contact with animals, with family, and so on. And our genetics. But genetic doesn't seem to be the main factor, at least for the global north. So the consequence is that when we are looking to larger numbers of people, that is possible now, we can see in this study that was published in 2023, where they analyzed 35,000 of microbiota from different countries, that you have a stratification of the microbiota. You can see that it's not dots all around. There is some structure in the gut microbiota. And it's what we call enterotype, and they are represented by this blue, yellow, purple, and green color. But then you also have those what is called enterobranches, or different subtypes. And they are quite linked to the type of diet and food consumption that you have. Okay? So it explains, at least in part, why we are all similar. We all have a microbiota, and we have some species which are well spread, well shared between us. But for most of us, we have a microbiota which is more than 90% different from another person in this room. Using those large numbers also allow to re-examine the signature, microbial signature or metabolite signature, which are associated with health or disease and with diet. And in that study with 34,000 of people from United States and United Kingdom, they identified 30 species which are signature of health for the 15 which are on the top here. And this considering different cohorts, so different studies, different population, and 15 signature which are associated with bad health. Okay? So it's really important now to have those big numbers to try to identify these biomarkers. For very long, the field was marked by numerous studies saying, hey, I studied 50 people and I have a biomarker for colorectal cancer or for healthy diet or for, And of course, it was not true, because another study with 50 people, even in the same country, will not find the same type of conclusion, So taking into account the variation of people and the variation of lifestyle, we need to have those big numbers. Still, in that case, those biomarkers, so it might be interesting, but the robustness are still to be established. And it's also one of the problems now in the field, is that you have now very large number of publications about biomarkers and possible candidates for solution, microbial solution or metabolite solution, but still the demonstration of the robustness of the biomarker is often not done. So your talk was were very important, because I think we're missing a lot those kind of rational validation of biomarker at this stage. So of course we know much more now about diet, and you see here kind of 6 typical diet, from Western diet to Mediterranean diet, where we know. So here you have the more or less diet raw composition in terms of lipid, carbohydrate, protein, or alcohol. We know what is the effect, the main effect on the microbiota. We know what are the main effects on metabolites, and we know how it modified the host, response or physiology at some level at least, But so if we looked a bit more to this Western style diet, so we know it's a diet which is not to our health advantage. What we see is that all the main food groups of the Western-like diet indeed affect the microbiota composition and function. And you can see that from digestible carbohydrate to saturated fat to animal-based protein or food additives, all of them, through different pathway or different modification of the microbiota, lead to gut barrier dysfunction and mucosal immune perturbation, not saying dysfunction. Okay? So we have those kind of, for some specific well-defined condition, we have mechanistic information on what's going on. Then some other diet seems to be much more healthy. And one of the really, I think, well-known example is the Mediterranean diet, which was quite studied, mostly, a lot by Italian colleagues, and that's very important. And you see here kind of summary showing that this diet reduces the incidence of chronic diseases and some cancers. It's not all non-communicable diseases, but quite a number. And overall, it reduced by 20% the risk of premature death from one of those diseases. So, which is quite significant. So, of course, we have diet to maintain health, but it's not only the Mediterranean diet, because we cannot all eat Mediterranean, So we have to now to design, to understand what's happening with the Mediterranean diet and what are the mechanisms behind, and how we can translate the advantage of Mediterranean diet to, for example, the new Nordic diet, Atlantic diet in Africa, diet taking into account also the local production, the cultural habit of people. And so it's really quite a work which is still ongoing. Then, however, there is also other type of diet which have been elaborated, mostly by nutritionists. So meaning that we don't always have information about what's happening at the gut microbiota level. And for example, one which is very well known also is the Heat-Lancet Commission diet, Heat-Lancet diet, which is based on nutritional information, but also on the objective of reducing the impact of our food consumption on the environment. Okay? And then you have more, so which is not the Mediterranean diet, but has similar trends, meaning lowering the meat, and especially red meat, and increasing very significantly vegetable consumption and fruit consumption and pulses consumption. Okay, so then the question is, beyond prevention, can we use diet for therapeutic benefits? Okay? So, yes, I see the time. So, of course, the rationale behind is that diet affects not only the gut microbiota, but all the body systems, such as cardiovascular system, muscle and skeletal system, endocrine system, metabolic system, and so on. So there were very numerous intervention trials in different conditions, but most of them are limited in terms of number of subjects. So it's difficult to conclude from one trial to another what will be the similarity, and often the intervention design are different. So there is really a need there for harmonization and standardization. Still, what we can see, so we already mentioned Mediterranean diet and the benefit on targeted disease, but the DASH diet, for example, which is low salt, was really designed to fight against hypertension, and just by the diet, it has equivalent result as monotherapy in that field. Another example is type 2 diabetes, where dietary approach have equivalent result as the standard of care. Okay? So it's at least, and there is a number of others. So if we focus a little bit more on type 2 diabetes, as you see, there have been different approaches, different intervention, but what is seen is that the effects are quite positive, from equivalent to standard of care to 60% remission. The main difficulty in that case is the observance of the diet over long term for the patient. Still, even if 61% remission is good, you still have 39% of patients which doesn't have solution, and this is probably linked to the variability of the people. And it's why now I think we enter a field where we cannot speak only about general effect of diet or microbiota or host, but we really have to go deep inside and to understand those variabilities and integrate them in our approaches. If we look at inflammatory bowel disease, so it's an important disease, and here we had 160 clinical trials with dietary intervention, but what happened is quite different from type 2 diabetes. If you consider pediatric IBD, then there is one solution which is exclusive enteral diet, and nobody understands why it's so successful today, And it's basically resetting the system using a very simple diet, It's no question of fiber or what. It's just giving food to the body in the simplest form. Then, for all the other attempts, we are on around 20% of success, which is very different situation from that of type 2 diabetes. So we still have a lot to learn. So to conclude, because I see that Juan and Moez are getting nervous, we are moving from small studies to analysis of data from tens of thousands of individuals, and it's good because it's making things much more robust. Still, we have to remember that there is a geographical representation bias in all the data, because for example, a few years ago, 2024, 70% of the metagenomic data corresponded to 15% of the world population, mainly Americans and Europeans. So it means that the rest of the world have very limited data, and as we show with diet, but also cultural habit, acceptability of solution, and so on, it's really important to have information on those countries. Prevention, nutrition is an important factor, but we still have gaps in the use of knowledge among the population, particularly among young people. I think, I have no views. They are, teenager or a little bit above. and they have disastrous nutritional habit. It's just terrible for me to see them with their Coke and you know, but you know, it's life. But we have to understand why, you know, and to see how to combat them. Therapy. It's an area of research which is going to really develop. There is lots of much interest for it. There have been successes, but there is still much to understand, and I think it will really develop in the future. It's quite interesting, and I think we should consider 2 points. In most of the clinical trial, people are focusing on the physiological parameter which are associated to the disease. But diet is a major confounder, which is most of the time totally forgotten. And the second point is that here, really, we have to deep inside stratification and to understand what are the subgroup of patients if we want to build solution. So more personalized approach are essential for the future. And just to conclude in a broader context, you know, we have these links between environment, food system, food demand, dietary pattern, and health. And what is good is that if we switch to healthier diet, we're not only going to have better life, but we are going to contribute to the decrease of NCDs around the world. We are going to modify the demand and the increase of demand in food because it's a more reasonable diet, I would say. And we are going to reduce the impact of the food system on the environment. So benefit everywhere. What can I say? And I thank you for your attention.
Thank you so much, Emmanuelle. I think we have time for two. Okay. Yeah.
Question. So do you think that the relationships between nutrition, microbiota, and health are well studied today? And maybe another one, what is the next step to engineering the microbiota for better health?
Good questions. Well, I think that it's difficult to answer the first one because it's changing with knowledge. So something which is considered very relevant when you have some level of knowledge about the system, after a few years, if the knowledge really increases, it's becoming irrelevant, you know. But still, it was important to go through that. So, for example, if we go back to those trials and the number of people, you know, it was really important to have small trials with a number of people showing that they can have an effect, you know, to convince people and to be able to go for more information, you know. It's very important to have information about what happened in, you know, high-income developed country because we can share those examples with people from lower-income country and they can understand faster why is it important to look also to diet, to microbiota, to develop public policies in those fields, you know. So that was the first question. Second question?
Second question about the next step to engineering the microbiota for better health.
Mm-hmm. So there is lots of study on that, how to engineer microbiota, and which go from, you know, easy one, go for physical activities and eat differently. But it's very general recommendation, and indeed, when you talk to clinician or doctor, many of them are not so aware about that. And there is no, you know, we're starting to hear that for obesity, for example, we should have not only consider a restrictive diet, but also, you know, have really understand what's going on, having psychological support, having, you know, exercise and so on, physical exercise and so on. But in practice, except if you go to very few centers which will, you know, propose those kind of approaches, you will be left with, okay, you should be careful about what you eat and stop chocolate or whatever. And we know that it's much more complicated than that. So I think we still have a lot to do. And regarding engineering of microbiota, I think we don't have the solution yet. You know, there is studies on how phages can modulate the gut microbiota.
Yeah.
There is studies on how CRISPR-Cas9 can modulate some of the pathogen or pathobiont present in the microbiota. There is those rationally designed consortia, how they can complement the microbiota. All that is under study yet, but I think now today we don't have the solution that will be universal and so on. And there is probably no universal solution. I would suggest that I think that probably we will have to adapt to specific condition or disease or people and specificity. Yeah.
Okay. So again, thank you so much, Emmanuelle. Thank you so much.